We will first talk about the definitions and the type of gene therapies, and also give you some examples. And then Jennifer and Carlo will discuss CMC and non-clinical considerations, challenges, and they will try also to provide you some suggestions, insights on how to overcome these challenges. And finally, some benchmark and trends for the future, followed by Q&A.
So those of you based outside Europe or the US might not be aware of the terminology used in these regions. The IATMP denomination is used in Europe. This acronym stands for Advanced Therapy Medicinal Product. The reason is probably because this kind of product is considered superior or first class when compared to other medicinal products.
So whether it's a raw, ancillary, or a starting material, your process input must be of appropriate quality, as any impurities present in these materials may ultimately remain in the final product. All input materials must be considered in terms of their risk of introducing viral contaminants to the process. And as a best practice, manufacturers should avoid the use of any animal-derived materials as much as possible and use materials manufactured in a GMP facility to ensure consistent quality and control of those materials.
Okay, one thing that you should always remember is also the 3 R's rule. Reduce, refine, replace, in order to minimize the number of animals used in development. This is certainly valid in EU and US, maybe less in China, but you need always try to do only necessary studies.
You provided an example of potency metrics for a vector-based product. What is an example of a potency metrics for an ex vivo modified cellular-based product? Maybe it's for you, Jennifer.
Yes, thank you, Sunji. So if we could consider, for an example, a CAR T product expressing a cytokine. So your potency matrix would, of course, need to be phase dependent. So you could consider transduction efficiency, percent CAR positive cells for early potency, and also transgene expression for your early phase potency matrix. And then as you proceed, you need to show more functionality. So in this example, that would include secretion of the cytokine, so an assay to measure that, and also ultimately like a cell killing assay, depending on your mechanism of action of the product.